the biggest problem with idiopathic pulmonary fibrosis and pulmonary fibrosis over the years was that most of the things we knew about the disease were from lab work on animals, on mice. So many of the mechanisms and hypotheses that the community has followed came from, again, laboratory animals, which is completely okay. This is the way science is. But you have to understand the human disease to develop therapies and potentially cures. In the last, again, 10, 15 years, there's been a lot of progress in understanding what we call the genomics of the disease, how genes and molecules change in the patients with pulmonary fibrosis. And many of the things we know now come out of that. In the last 3 years, we've had basically an amazing leap, an amazing breakthrough, which is now we are not dependent on the analysis of the organ. Our resolution is very, very high. So if you think about pulmonary fibrosis is basically a disease of an organ. The lung is getting scarred. But what is the lung? The lung is basically a collection of cells that are organized in a way that allow them to do respiration. Cells are the smallest units of the living organism. And in pulmonary fibrosis, these cells, for a variety of reasons, start behaving differently. We get collection of new cells coming in the lung. How would we say, your good old functional cells starts being bad for you for some reason. It is not cancer, but it's very reminiscent of what happens in cancer. So now with new technologies, we can actually profile every one of these cells. So my group and other groups have been collecting human samples and profiling every single cell. So we could take a person and we will profile hundreds of thousands of cells. And this is just the initial phase of it. The first study was finished a year ago. My group just posted our data. This is publicly available. So we created a website that is mostly for scientists and physicians, but even for patients and family members. You can go in on the website. It's publicly available. It is IPFcellatlas.com. And basically you can look there and you could look whether a molecule is changed in a cell and whether the cell population changes in the disease. Now, the reason that this is important for us is two things. One is the resolution of the findings allow us to understand that most of the drugs we use now are sort of only hitting the biggest impact, the sort of the edge of the iceberg. And as you know about an iceberg, everything is actually below water. And with these new tools, we're starting to understand what is the things that we didn't see about IPF, what we didn't know. How can we change? Because really for me, personally, and for our patients and their families, slowing down the disease is not enough. We want to stop it. We want to reverse it. And the only way we'll be able to do it is by understanding how every cell in the fibrotic lung speaks with another one, and then target a molecule that basically fixes the cell. And we're very excited about it. The other thing I want to mention is the importance of data sharing and data dissemination. So the reason I mentioned this, and the reason we put in the data public for the whole world, even before we publish a paper, so even before we secure the rights, is because we want to accelerate discovery. We're dealing with hundreds of thousands of cells and tens of thousands of genes, and I don't think that I am that smart to control all of this information. Also, if I wanted to study every one of these elements, it would take me 100,000 years. But by just putting it out there, I hope that 100,000 people, investigators, smart, knowledgeable, will get interested in pulmonary fibrosis, share their insight, and basically create, again, what we said before, this critical mass of investigation. And really focus not on low-hanging fruits of slowing down disease, but on the biggest ambition, which is, cure pulmonary fibrosis.